coming. It's indeed my pleasure to have Mihai Pop kicking off the Fall Colloquium series. Apologies to those of you who couldn't be here today because of Rosh Hashanah. So that's one of the reasons why we are taping the talk. So happy Rosh Hashanah if you watched the video. Mihai has been a wonderful colleague here for a number of years. He got his PhD at Johns Hopkins University, doing some stuff working on my end of the world. He worked with Professor Rao Kosaraju at Johns Hopkins doing geographical information systems and algorithms. And then he joined Stephen Salzberg working at Tiger, doing a lot of genome analysis. And he's been a member of UMIAC, CBCD, NCS for a number of years. And his work has sort of evolved over time. He started off doing mostly genome analysis, and now he works in the microbiome lately. And there's a number of software packages that the group has released. I just want to mention one, Bowtie, which is quite well regarded. And it's indeed great to have Mihai speak to us today. Thanks.
Thank you. Yeah, thanks all for being here. So I can't use my laser pointer on this, so I'll use my hands. Hopefully that will be okay with everyone. To frame my talk and the kind of things that I'm working on right now, I want to kind of pose one of the application areas that we're focusing in. So these are some pictures taken by one of the students that I co-advised in Calcutta, and this is in some parts of Calcutta how you get your drinking water. So clearly fairly unsanitary, and the result of this is that there are lots of instances of diarrheal disease. And in general in the world, this is something I didn't know before we started the study, was that diarrhea actually kills more people or more children than HIV, malaria, and measles combined, right? So it's not something you hear in the news all the time, diarrhea, you hear about HIV, and you hear about malaria, you hear about measles, but diarrhea is actually much more deadly. So, and this is the area, this is one of the studies that we got involved in, trying to understand a little better what happens with diarrhea. And because I'm a computer scientist, I'm going to explain to you what computational challenges we ran into. But I also want to stress that this is probably harder than proving any theorem. Solving this problem is harder than proving any theorem or generating any algorithm in the world. So, and those are the kind of problems that motivate me. The one thing with diarrhea, and this is something that if you are any biologist says, well, any doctor probably, I don't know if any biologist, but any doctor would say, I know what causes diarrhea, and they can immediately list a bunch of names of organisms that cause diarrhea. And that's all and good. Our colleagues at School of Medicine tried to develop started a study to try to understand a little better. Well, we know what causes diarrhea. Let's find out which of those causes are the most important. Perhaps we can, if we know that norovirus is the most important, at least it is the most important for Disney cruises, right? But it's we can figure out perhaps how to develop new vaccines, how to develop new treatments, and so on. So they started this study. It's actually an amazingly large study. So they looked at 22,000 children in 7 countries. Out of those, we looked at 4 of them in more detail, and I'll talk a little more about that in a second. But this is an extremely large number of children, especially as they had to collect samples. Oftentimes they had to go on a motorbike on a dirt road to get to a small village to find a child and collect the samples and so on, and then process them. It was really an amazing feat of organization, and so it's just amazing study. What they found out after looking at all these children was that in over half of the cases of diarrhea they looked at, they could not identify which pathogen caused diarrhea. Right? So we know so much about diarrhea, yet 1 in 2 children were sick, and we had no clue why they were sick. And this is running the traditional tests that someone would run at a hospital. So it's equivalent to you going to a doctor, they run a test, and half the time say, I don't know what's going on, So this is a pretty clearly we don't understand everything that we hope to understand about diarrhea. This is what this is for at some level why I jumped into this project. And I want to explain that biology is fairly simple, so you can explain what biologists want to do with Common Core standards. So, and these are the Common Core standards from first grade, which I received recently. So, right. So one of the things you do is you compare by identifying similarities and differences, and you sort and classify into categories. Right. So this is when you're studying diarrhea. This is what you do. When you're studying biological instances, this is what you do, and people have been doing this since the 17th century, right? So this is a picture of the first microscope from Anton de Leeuwenhoek, and this is what he could see, right? He could see little things, and he's already, I mean, this is from the 17th century. He already figured out what you want to cluster things together based on how they look or how they move and that kind of stuff, right? So that was 17th century biology. It's probably a large part of 21st century biology as well. Right? But a lot of 21st century biology now has slightly different microscopes and slightly different pictures, right? So this is what we see today. And we don't just see maybe a couple hundred dots in a little plate. Now we can see tens of millions, hundreds of millions of sequences that look like this, okay? So now the question is, well, how do you do this? How do you answer this basic question that first graders are learning how to answer, like what's same and what's different? How do we answer it with computational tools? Okay, that's the problem. One of the first problems we have to tackle. Okay, so first, before trying to figure out exactly how we answer this question, you have to sort of understand a little bit where the data come from. Okay. So, so it's not just them giving you pieces of DNA. There's two different ways of getting pieces of DNA from bacteria. So then we can look at those sequences and try to understand exactly what's same and different. So say in both situations here, I have a red organism, a blue organism. In the first case, I'm simply looking at a single gene, sequencing just that gene, and asking, well, are those sequences from the same place in the genome, from different genomes? Are they similar or different? Right. So this at some level is a fairly simple operation that I have to do because I know exactly all those sequences come from the same exact place, and I can compare them directly to each other, right? So I can tell you that there's two red sequences and one blue one, presumably fairly easily, right? There's another approach I can take, and that other approach involves just sequencing randomly. You take the organisms that might be in a sample, you break them apart, you generate sequences from them. And that's what people usually call whole genome sequencing. And if you do that, you're going to get, say, the same three sequences as you got here, but now there's no way to know that these two sequences at the top come from the red organism. They could come from completely different organisms. There's no way to connect them to each other, right? So the question is, how do we deal with this? And the way we deal with this is by generating many more sequences until we can reconstruct those organisms. So now we have what we call an assembly of the two organisms, one that's red, one that's blue, and now we can tell, well, look, that this sequence at the bottom is really related to all the other sequences that form the blue organism. The two red sequences at the top are connected by many other sequences, so they belong to the same organism. Right, so now we can answer the same questions. So this is another area of the work that I'm going to talk about, another area of my work. But first, a lot of what I've done has focused on this data, and I don't know if anyone has a guess why most of the data comes from experiments like this. Anybody?
Money.
So, I'm sorry?
Money.
Money? It's exactly money, right? So here I generate three sequences, I can tell you there's two organisms. Here, to tell you there's two organisms, I have to generate several hundred sequences, right? In reality, the difference in cost, and we've costed this for grants and so on, is a factor somewhere between 10 and 100 in terms of the cost of an experiment run this way versus an experiment run this way. Or with another way, you can sequence 10 to 100 times more samples with the same amount of money if you do it this way than this way. So that's actually the only way we could do that study on diarrhea. We only looked at 3,000 samples, but even there we needed money not from the NIH but the Gates Foundation to get enough money to do the sequencing for that experiment. We wouldn't have been able to do it this way at all. Okay? One of the problems, I mean, even though I'm saying that this problem is easy, one of the big problems that arises when I have something like, say, 3 million sequences I want to compare to each other is that if I really want to find out how they cluster and so on, I have to do at least in theory, if you think about it, I have to do 3 million times 3 million operations, right? So I have 9 trillion operations that I have to make in order to build clusters of the sequences. And each operation is what we call, well, it's usually, well, the way I think it should be done, and we all agree, but essentially you have to do a full dynamic programming to do a full alignment between the two sequences. So that operation itself is quadratic in the size of the sequences themselves, okay? So essentially you have a very expensive operation generating each of those edges, and then you have something that's on the order of n squared, different edges that I would need to process in order to build a cluster. Needless to say, this was taking quite a bit of time on a computer, so we had to figure out ways to do it faster. And I think you have to give up some stuff, and I'll mention that. But essentially what we end up doing is use a clustering heuristic that's fairly simple. So essentially this heuristic says, well, take a sequence. If it has not been clustered already, it's obviously now going to form a new cluster. And then search through all the other sequences and recruit any sequence that matches that sequence, and now you form a cluster. Okay, so each of these, so essentially I have this represented, I'm just fishing out sequences to make its own cloud of sequences. And when I'm doing this, each of these edges again is one of these operations that's full dynamic programming. It's a fairly expensive operation, but now I only have to do it between the sequences that become clusters and all the other sequences. So it's no longer n squared. It's number of sequences, number of clusters times all the number of all the sequences, right? On top of this, we can actually be a little smarter about that, and I'll get to it in a second. But why I like to do this and with the full dynamic programming is that sorry, Samir?
So when you take two sequences, how do you define the exact distance between them? Is it just the edit distance?
It's the way we define it is the edit distance, but you can add scores to allow certain types of edits to be more costly or cheaper, that kind of stuff. But what we implement is pretty much plain old edit distance. Yeah. So anyway, so we. Right, so the reason I'm using full edit distance, and a lot of biologists don't, know that computer scientists work with biologists don't for a variety of reasons, but I'm using that because I can give you a guarantee. I know edit distance as obeys triangle inequality, and I can tell you that my clusters will follow the property that for whatever sequence I picked, all the other sequences are within some distance from the center, and they're within some other distance from each other, right? So it's at most twice the distance from the center from each other. With a little bit of tweaking of the algorithm, you can also guarantee that any sequence gets assigned, like for any sequence, the center of the cluster we picked is actually the closest sequence to it, okay? Which you can't guarantee if you do plain old algorithms. I like the idea of being able to tell you, sure, I'm doing something heuristic, but here are the guarantees I can provide, okay? And I think this is important, and at some point it will be important to understand if those guarantees are what the biologists really want, but that's a little more complicated. The other stuff I want to mention is the search, right? So I have to search through all the sequences, and that could be extremely expensive as well if I'm doing dynamic programming. Like each of these edges could be a separate dynamic programming. Instead, what we do, we organize the sequences in a trie. We don't actually build the trie itself, we just sort the sequences. And it helps a lot that all the sequences get aligned and the left end the way the experiment is run, so we can have that guarantee. But if the sequences are organized in a trie, whatever query sequence, the center that I'm searching against, I can build a dynamic programming table that kind of follows the trie. And when you get stuck, you backtrack, and you don't have to recompute. So all this part here, that whatever query I have, I'm searching against the common prefix of all the sequences. I build a dynamic programming table, and now as I extend to the right, I just need to fill in the blue part of dynamic programming table. When I'm done with that, and I backtrack, and I follow another path, I simply revert to here, and I have all the information already computed, right? So essentially, the runtime is no longer n squared, if you will, with respect to one query against one sequence in my database, but in the same operation, I actually handle all the sequences that have the same prefix. If there's lots of them, I get huge savings in runtime. Okay? And that's what we've been doing, and this was work that my student Mohammed Ghodsi did, and he's now at Google. So we did that, and for the first data set, we're quite happy. We got great results. And then we tried a new data set, and then now suddenly things didn't work quite as planned. So we calculated at some point as the algorithm was running, that would take us about 3 months to get the results. And that's not something that biologists are willing to wait for. Okay, so we try to understand a little better what's happening, and this is sort of an unsolved problem still. I'm trying to work on it a little more with an undergrad right now. But the basic idea of what we tried to do, we said, well, it looked like the runtime was still quadratic, right? We think we're saving a lot of time, but the runtime was still quadratic. So to better understand what's going on, what we did, we took a subsample of the data, clustered that subsample. So say we take square root of n of the data, quadratic is still linear time to compute the clustering of just that subsample, right? And then any sequence that fell in a cluster now became the center of a cluster that I can just search against the rest of the database and fetch it in. So that essentially, in essence, what we're doing, we're taking the abundant clusters and starting with those, right? Because if in a subsample something clusters, then it's probably part of a larger cluster, okay? So what we did there, and I'm not sure how to best represent this, but essentially we started with 30 million sequences, and we ran this clustering, and about half of them ended up in clusters. We ran through this process, and it happened quite fast. So the estimate we had was about 236 sequences could be clustered per second. Okay, so this was not a ridiculous amount of time to cluster half the data, right? So like, well, okay, this works really well. We'll take the half of the data that was not clustered and repeat the process, right? What could go wrong? So when we did the second half, we managed to cluster another 1 million sequences. So not half of what was left, but 1/16 of what was left. And obviously the clustering speed went down. And then the third part got even less and even less. So essentially we got very quickly to this point of diminishing returns. So what it turns out that's happening, and I think this is an example of this curse of dimensionality, we have a lot of noise in the data. And essentially, it's very easy for us, for a sequence that's in a large cluster, it's very easy to find what the cluster is. For a sequence that's unique, because it's got too many errors in it, we have to search it against all the other sequences before we can decide that that sequence is unique. Right? So you can do some math, we can come up with, for sequences we have a length 500, you have 95 trillion sequences that are just beyond our limit of similarity that we chose, that we can construct with like simple edits from that sequence. So essentially, we really can't escape searching everything else before we decide that sequence cannot be clustered. And we have to do the full dynamic programming all the way to the end. So the reason I'm saying this is kind of an unsolved problem is that I still want guarantees. So I'm fine having this algorithm. I run it, half the sequences are clustered, I'm done, I'm using those clusters, do analysis on them and so on. But I want to have a guarantee that I'm not losing, that the other half of the sequences don't contain clusters that are interesting. And I can't come up with a fast algorithm that will actually get me that guarantee. So I'd like some kind of probabilistic guarantee to that. Like with high probability, any cluster that's more than, I don't know, 10% of the data set will end up being in my data set. So, so if any one of you is interested, I can talk more about that. Okay. Coming back to the Common Core standards, right? So the next question is, what do we do with these clusters, right? So I'm not just building clusters because I like building clusters. We're building clusters because we want to understand something about diarrhea in our case, right? So essentially, what we're trying to do is we're building a matrix that looks like this, where I have, say, the healthy and the sick children, and I have a set of clusters, and here I'm plotting a heat map of the abundance of those clusters. Like blue is high abundance, red is low abundance, right? So what I really want to be able to say, oh look, there's a set of clusters over here that are all really abundant in healthy and not abundant in sick. So perhaps these are things I can give, probiotics. I can make yogurts with this, give to the children, and they don't get diarrhea, right? That's the kind of stuff we might want to do. So the question is how we do this, huh? And how we do it the right way. And I won't give you all the answers because Hector will give you all the answers soon. Perhaps. We haven't coordinated. But essentially, there are a couple of problems that we have to deal with. So first of all, if you think about the cluster that's present in it's about 10% of your data, right? If I've sequenced 4,000 sequences, I would expect about 400 to be in that cluster, right? So that would be the abundance I observe for the cluster. If I sequence 10,000 sequences, I expect about 1,000 sequences to be in that cluster, right? So clearly the amount of money I throw at the sequencing doesn't change the abundance of an organism, but it appears to change at least computationally, right? So we have to correct for that. We have to get rid of the dependence on the count or the abundance of any organism we see from the dependence on the amount of sequencing that we did, right? The second part that we have to deal with is the fact, and this is what this plot is showing you, is that the more you sequence, the more you see. So it's not just that the abundances change, but also the number of clusters that we see in the data change, right? So suddenly there's organisms that appear because they were before below the limit of detection, right? So, and what we want to do with this mixture of signal, we want to do an association study. We want to ask statistically whether there is a difference, whether the difference between cases and controls can be explained by the abundance of those of what you call cluster, right? So, so addressing this, and this is a very nice model that Hector came up with, was essentially to start to model the fact that a particular sequence could be absent from the data. Just because of undersampling, to model that inside the statistical model that does the association statistics. Okay, and I won't go into the details of this again. Again, Hector will show you that, but essentially what we show is that the results that we get in terms of the AUC, essentially the ability to classify healthy versus sick with these sequences, or to identify a particular cluster that's differentially abundant, they outperform all the other methods that are used in the field. Okay, and this is, and we believe a lot of it is because of exactly modeling, specifically modeling the zeros in our data as being either absent or absent because of undersampling, which have different properties statistically. Okay, and again, this was work that appeared in Nature Methods, and Hector will probably talk more about it. Okay. But I want to say the one thing that I want to kind of like a take-home lesson, especially for the younger people in the audience, is that you know, both clustering, association statistics, all this stuff, these are problems that have been addressed for easily 20, 30, 40 years, right? If not more. So essentially, the key question of making some success or actually making new contributions to those fields is actually understanding what the biologists want out of the data. Right, so if you ask me, if you had asked me, well, build a clustering algorithm that's better than other clustering algorithms out there, right? There's hundreds of papers on clustering with different assumptions and so on, and it'd be really hard to maybe shave a log n of some algorithm or get a tighter bound in approximation and stuff like that. But in this case, we actually figure out what biologists need out of the data and could develop something that addresses the biological question, even if perhaps on the computer science side it doesn't actually give you a huge improvement. And I don't know if it does, but it's a very specific problem no one has looked at before at some level. So we have room to make contributions. And this also, this is a really hard part of doing work in computational biology, is understanding what biologists want, not what we think they want. And so if those who find that secret, that then have a great collaboration with biologists, right? If you don't find that secret, then you develop, you write papers, but not make contributions to biology. So just to give you a little bit of an idea of whether we found something new, right? So essentially, we started all of this not because we want to develop clustering algorithms or association statistics and so on. We did it because we tried to solve diarrhea. Okay? And we try to analyze the data coming from this study to better understand what's going on in diarrhea. And we could see, in this case, we can separate out the plots based on case and control. So the case would be the children with diarrhea, without diarrhea, and their ages. And it's not necessarily easy to see, but there are differences, especially if you look here, there's a lot more green in the cases, a lot less green in controls. Also, you can kind of, and that's one group of organisms. In this case, I think that they're Escherichia organisms, which are E. coli. We know E. coli causes diarrhea, so that's a nice finding there. And we can also see how the diversity of the gut microbiome changes over time in children. And this was kind of interesting. Essentially, we start with a very low diversity environment, and then it grows very rapidly between 0 and 12 months of age. So in the first year, we develop what's called normal flora in our guts. And also you can kind of see that the flora looks pretty bad in the sense that you have a lot of kind of E. coli type organisms that they quickly disappear as you get older. And that actually turns out to be very important for child development. The fact that you're attacked by pathogens early on allows you to develop an immune system in your gut, which otherwise you wouldn't be able to develop. But anyway, so we've learned some things about this. The most interesting thing that we learned was that there's a group of organisms called Streptococcus. I mean, you all know strep throat, for example, right? And no one, they're not on the list I gave you before, what causes diarrhea. No one thought they caused diarrhea, and yet in our study they come highly associated statistically with diarrhea. So we have a new hypothesis that streptococcus, or at least some streptococci, may cause diarrhea, and now that's something that allows us to develop new tests to find the streptococcus and perhaps even develop a treatment against them. The one thing you should realize is you shouldn't just believe something because a computer told you it is so. Definitely biologists don't believe it. So our biology colleagues went to did experiments, and here's an experiment looking at human epithelial cells. This is the response that you get to different bacteria. This is E. coli, we know it causes diarrhea. This is an uninfected, so it's actually green response means there's no diarrhea. Red response means diarrhea, and there's a streptococcus strain that actually exhibits the same behavior as a diarrheogenic E. coli in this cell culture experiment. So in this case, our predictions, computational predictions, were confirmed in the lab. Okay, and we're chasing some of these down. Okay, the second part I mentioned that I do genome assembly, right? I want to reconstruct genomes. So this was. Up to this point, I was just focused on one gene because that's the data that we had. But now the question is, what do we do when we're looking at mixtures of genomes? And I'll give you a very quick survey of what we have to do when I'm talking about genome assembly, right? So this is a great example. Two of our students came up with this. Adam Philippi and Michael Schatz came up with this example. We take the tale of two cities, break it up into small pieces, and try to put it back together. Right? So it should be easy. It's in English. You don't need to know any biology to do this, right? So it's fairly easy to actually reconstruct it by hand up to a point when you realize that the problem is a little more complicated than you initially thought because it's an ambiguity. If you're looking at times it was it could be times it was the age, times it was the worst, times it was the best, and so on. So you don't really know what you have to do. And the common paradigm being used today in genome assembly is to represent everything as a graph and try to think more globally about the graph structure. So you're not sure if you should take the path to it was the age or it was the worst, but you realize that if you take the path to it was the age, you have to skip all this loop. So essentially parts of the genome remain unassembled. So clearly that's a bad choice. I'm going to take this path first and then go to the it was the age and solve the genome assembly correctly, right? So this is all very well, and Pavel Plesner will tell you that now the problem is completely solved, so we're done, right? So it's actually a pretty simple problem. The reality is that this is what you see in a real genome. So this is a single genome, and I'll get back to multiple genomes. This is a perfect assembly graph for a real genome. It's actually a very simple genome. And each node is colored by the number of times you see that node. So green is 1, blue is 2. Orange is 3, and red is 4, right? So I don't even have things that get repeated multiple times. I mean, 4 is the largest times I see something in the genome, and this is the picture that you get, right? So obviously you can't solve this. I don't know if it's obvious. I can, you can't solve it even on a computer, right? You can find a tour through this, because this is an Eulerian graph. You can find one tour, and you can find it efficiently. But that tour might not be the correct one because there's an exponential number of other tours, all of which you can find in linear time, one at a time if you wanted to, right? But only one of them is correct. So now the question is, how do you actually find a tour that is correct? So it's not an easy job. So we tried to figure out exactly kind of what the limits are, and this is work that was a little more theoretical, just to try to understand what's difficult about the assembly, because in part, I'm telling you that this is essentially impossible, right? I'm giving you this, I'm telling you, you can't computationally solve the problem, right? At the same time, if you give biologists the biologists data, shove it into a program, it gets outputs, and they say, well, look, the outputs look fine. So assembly is easy, right? It only took me 5 hours and that's it. It's not a big deal, right? So we try to understand exactly what's going on, and it turns out that you can actually prove things about what makes the assembly problem hard, and you can roughly summarize it in the assembly problem being in 3 different categories, and depending on the relationship between the length of the sequences that you generate in your experiment and the repeats in your data. So if the reads are much, much longer than the repeats, then you have no problem. Then anybody can write a shell script to do assembly, okay? And people have done that in the Human Genome Project. If the reads are much shorter than the repeats, then you get into the situation where you have an exponential number of genomes that you can reconstruct with the same data, all of them being equally good as far as the fit of the data to the genome, right? So in that case, essentially, it's an ill-posed problem, right? I don't have a solution, right? I've just chopped up the genome so much that I no longer have one solution. I have an exponential number of solutions. So I don't really know what it means to really solve the assembly problem in that setting. And then in between, you get into this kind of strange zone that we as computer scientists like so much, which is the empty hard state, right? So this is where we know there's a single solution, but we can't find it efficiently, right? But this situation actually occurs fairly rarely, and most of the time you're either here or here. And that's why biologists would say, oh, the problem is easy to solve, because for large chunks of the genome, this is exactly what you're looking at, and you get large chunks of the genome correctly assembled and being able to use the data. We've also looked at, so we've looked at what you can do if you have in this problem, in this situation, right? If the assembly problem is undetermined, there are many possible solutions. How do we know which one we want to pick? How do we focus ourselves towards the biologically correct version of the assembly? So one thing that we have, and this is where you have to go talk to biologists and find out what additional experimental data they can give you. So they don't just give you the fact that here are a bunch of sequences, but they also tell you these 2 sequences, the sequences come in pairs, and these pairs are a certain distance apart. Okay, so this sounds like a very valuable information, right? So what you can do if you're looking at a complicated graph, you can say, well, I know the distance between these 2 blue nodes, and I know that there are 2 paths connecting those blue nodes to each other, one that's shorter, one that's longer, right? Well, if only one of them is consistent with the length of that constraint, then I'm done, right? Essentially, I can tell you I have to follow the thick path in order to satisfy this distance constraint between the 2 nodes, right? Does this make sense? I hope, right? Of course, what also happens is a situation where you have these 2 red nodes, and the 2 paths between the 2 red nodes are equally good or equally fit the constraint. In that case, there's nothing I can do about it, right? In that case, you want these kind of connections that span exactly one of these ambiguous nodes in the graph. So essentially, there are ways to actually use this information to constrain the graph structure to something that makes sense, but they don't universally work, and we've tried a variety of things to see exactly what happens and to just demonstrate that these approaches don't always work. And here's a plot where we generated many different pairs of constraints, try to solve the assembly problem, see how far we got, close to 100%, which would be that the genome is correctly assembled computationally, and we could never get to 100%, okay, but just using this information. So there's still room for improvement. And we also did some work, and this was with a great student, Joshua Wetzel, who was an undergrad at that time from Camden. He was one of I apologize, I forget the name, Rajiv Gandhi's students, right? He came here, and we also played with the idea that if we kind of tune the distances to the actual structure of the graph, we can get better results. And it turns out that we actually were able to do that. At the same time, it's kind of meaningless because biologists don't want to do the experiment the way we tell them to do it. They want to do the experiments the way they used to do it. So no one will actually do this for us. So. But it was nice theoretically. The other thing I wanted to mention, and this is kind of, maybe I should have asked you before, but part of it is what do I do, how do I get more information, how do I use all the information I have to solve the assembly problem. But another part you think about is what happens if you don't have enough information. So by not having enough information, usually in our case would be I have not sequenced enough, I haven't sampled all the genome correctly, okay, or not completely. The first intuition, definitely the first intuition of biologists, not computer scientists, I don't know what the intuition of computer scientists is here, but the intuition of biologists is that if you haven't sequenced enough, the assembly will be correct, but there will be gaps in it because I just simply have not seen those pieces of DNA. Right? That's the very simple intuition. But what I argue is that if you think about the graph structure that we're using, if you don't have enough information, you're going to miss parts of the graph. And then the other parts of the graph will look like a very simple thing, a very simple solution. You just solve the problem and you're done, right? But you are losing information that there's ambiguity in the reconstruction, and you might reconstruct the incorrect thing. So you might get it was the worst of times, it was the age of foolishness, which is not what Dickens wrote, okay? But it's correct. It's correct grammar, right? So it makes sense, but it's not what you want, right? And I also argue, I mean, this will. A lot of times some biologists say, well, but I'll look at it and I'll know it's wrong, right? I'm saying if I can't look at it in English and say it's wrong, how would you be able to do it in DNA, right? So you can easily get fooled when you don't have enough information. So this is what brings me to the kind of stuff that I do, where I thought the other problem was assembling a single genome is difficult, right? Or perhaps impossible. So I've decided that I don't want to assemble a single genome, I want to assemble mixtures of genomes. Right? So this is what I get. If I get one of those diarrhea samples, there's hundreds to thousands of genomes in one single sample. I want to assemble them all. Okay? So because one is hard, doing them all at the same time, it should be easy. Right? At least that's my English. I also worked on protein folding at some point in graduate school, so I'm a sucker for punishment. Right? Anyway, this is a really interesting area because it's another one of those areas where you find a problem that the world's best experts have not thought of yet. Right? Genome assembly, people have been thinking about it for over 30 years. There haven't been any real new ideas in genome assembly since about 2001, 2002. Okay, so there've been small improvements, nothing major, right? No one has yet thought about how to do this with many genomes. You can't really formulate the problem as easily as a path traversal problem, a graph traversal problem, because now you're looking not for one path, but you're looking for many different paths that might intersect or might not, and you don't know what the objective function should be and that kind of stuff, right? So it's even the mathematical formalism is not established at this point. And also biologists don't know what they want to get out of this. They say, well, assemble my genomes, assemble my community. Like, well, what does this mean? And I've asked them when I give talks or tutorials, I ask them, what do you want out of the data? And most of the time they don't know. So that's kind of interesting. One great answer I got from someone is, I want to know everything. So I want to know everything too, but. And my belief is that all good problems are ill-posed or intractable. Okay. So what we've been doing in this area, we've been trying to start to understand how we can untangle the different organisms from each other, find repeats, and so on. So this is work we've done with Sergei Koren and Todd Ragnar Nolnga here, and Jay, who's now in my lab. And here's a very simple mixture of organisms. There's two different organisms that are quite different from each other, actually, Staph aureus and E. coli. And the graph joins them together. So what we're looking at for ways of actually identifying these nodes in the graph that can now separate out the genomes. And one of the ideas, this was the idea Sergei came up with and Jay's extending right now, was to think of this as a social network and try to identify the important nodes in the social network. So the nodes are colored by between a centrality measure, and you can see that the nodes that separate out the genome have high between a centrality. And now you can say, well, perhaps this is one way of identifying nodes that will now make my graph simpler. Okay? So we're still exploring that. In more detail. But I'm not sure if between centrality is the only measure we can use or the most appropriate one, but it seems to somewhat work. The other thing that you can find in these graphs, and this is a picture from a paper in PNAS, so we're not involved in this paper, but this is the kind of data biologists are interested in generating. And this is also an infant gut. And they realized there's two types of one organism called Citrobacter, and they're different and they vary differently across time. So they're two, you could call them the same organism perhaps, or they're the same species for sure, but they have differences between them. They both exist at the same time in the samples and they vary differently. So we want to understand how to separate things that are very, very similar from each other, but with small differences. And the assembly graph, they actually built by hand. For this paper, it looks like a series of bubbles, and the path to the graph represents one of those organisms. Okay, so so the question is, well, how do you know, they did this by hand, but we can't afford to do this by hand for hundreds or thousands, or even for more than a couple of samples at a time, right? So the question is, can we start uncovering this type of information by looking at the graph structure more globally? Okay, so essentially one thing that we've done, and this seems to be working fairly well, we're still trying to kind of untangle all the stories, is to use a decomposition from graph theory called the SPQR tree decomposition of a graph. So this essentially breaks up the graph in a hierarchical manner into tri-connected components. So, so each tri-connected component in some sense, and these are some of those that we can get automatically from the graph structure, represents some region where perhaps there's some variation between the organisms that have been mixed together in the same sample. Okay, so we can discover, and we can actually, it's not easy to see, but we discover the same pattern in the same data set that they had in the paper I mentioned before. We're still trying to really understand what's going on here, because we want to go not just understand there's a pattern or one of these tri-connect components, but understand what it means biologically, right? And then try to add annotations and try to actually display to biologists in a way that makes sense to them. But this has been really promising work, and this is one of the areas I'm working actively in. Running out of time, so I'll go fairly quickly here. So one of the things that you have to realize with assembly is, right, assembly is difficult. You can have mistakes in it and not even know that you have mistakes and so on. So a big part of genome assembly, or what we should be doing, is evaluating whether the results are correct. And there's surprisingly little work in this field. There was a lot of work in the early days, but essentially we're developing tools that look at internal consistency of the assembly to try to understand where there are places where the consistency will based on our assumptions about the sequencing process, tell us something's wrong, and flag those regions so we can possibly correct them. But we're also looking at ways of using essentially probabilistic generative models to essentially ask, I have an assembly, I have the data that went into the assembly, how good is the fit between the two, right? And that's the likelihood that these sequences would have been generated from the assembly by a model of the sequencing process will tell us how good the assembly is. And this actually works really well. It's an approach that works really well for comparing different assemblies of the same data set. And there's some issues with extrapolating beyond that, in part because we don't really know how to estimate the probability of a set of reads or of the assembly itself at some level. So, and the last part, I think it's kind of starting to push towards the future, and that's the most exciting stuff that's happening. So one thing that we discovered in the diarrheal data set was that if you use, this is using like standard epidemiological tests, you can realize that if I have two different organisms, so in this case I'm looking at rotavirus and Shigella, if I have the two different organisms in a sample, then the likelihood that you have diarrhea in that sample is severely increased. Right? So the way you read this is the amount of diarrhea that can be explained by rotavirus. This is the amount explained by Shigella. If you put them together, you'd expect this, but what you really get in our data is this. Right? So this tells you Shigella and rotavirus work together to make you more sick. Right? So we discover interactions between organisms. And some interactions are positive, like over there, and other interactions are negative, in which case we're looking at lactobacillus, and this is something some probiotics have, right? And Shigella again, and you'd expect this amount of diarrhea when you put them both together, but what you see is actually much less. So this tells you at least this lactobacillus could help reduce diarrhea. And this is something actually my colleagues at School of Medicine are chasing down. They're actually trying to understand what compound in lactobacillus might cause, well, might kill Shigella. Okay? We also looked at the networks of interactions, and these are not quite interaction networks in that diarrheal sample, but the network of correlation networks, so I'd call them, because we can't actually tell you if any of these interactions are causal. And what you see is that if you look at diarrhea versus non-diarrhea, it's not just that the abundances of the organisms change, but the connections change as well. So understanding the disease might require you to understand interactions as much as it requires you to understand how individual organisms contribute to disease. So that's one area that we're looking at. Together with Matthias Chung from Virginia Tech, we've been looking at OD type models. We specifically developed a couple of their models to understand these interactions. And the models we have work better than the models from other papers, but I can give you more details later if you want. Okay, so the question is, what's next for me? In case anybody cares. If you want me in the department for longer, you probably care. So I'm really interested in this idea of looking at the structure of the genomes, like understanding these patterns of variation, understanding how it's not just the genomes change, the genome abundance changes, but also how the genome structure changes across samples, and how mixtures matter. I'm also very interested in how interaction actually shape microbial communities, and this is a big frustration of mine in the field in general. Every time we discover something simple, like looking at abundance, they say, hey, this organism is more abundant in these samples than in these other ones. There's some biologist says, oh, I saw that on a microscope before. Or the saddest thing is a lot of times they say, well, Pasteur saw that on a microscope before. So, right? But interactions you cannot see under a microscope. So there's a huge potential of actually uncovering new biology by looking at interactions, because that's not something that's very easily done in a lab. The other stuff that I think is critical for getting everything to work is, we're generating software tools, and we want to make sure that those software tools work. But most of the time, the software tools we develop, the output of the software tools is the first time anyone has seen an output like it. So how do you going to know if that output is correct or not, right? So it's the same problem with climate change, there's many other problems, and I assume a lot of these difficult machine learning approaches and so on. You get an output, well, what does it mean, right? And if you have a software bug, and I have plenty of examples of papers in Science and Nature withdrawn because of software bugs, right? If you have a software bug, you don't know if the result is due to a software bug or it's truly a new discovery, right? So the question is, how do you do testing? I'm working with Atif Memon on this and trying to figure out ways to test software that can be done without actually knowing what the answer looks like. The final thing, which probably should be the first thing, I think that we're at the university. I'm really interested in education and trying to improve 131 among other things. But just trying to figure out how to best teach not just students here, but also teach biologists about the computational concepts, not about how to use tools, but how to learn what those tools are doing and be able to make an informed decision of what tool to use. So I think that's it. I also have a strong belief that if you put computation and biology together, you get much better results. And I want to thank everybody who contributed, both my lab, the environment at Maryland, the CSU, MIAX, the CBCB, and so on. Also, I've been involved in a number of large projects at the NIH that have been really, really useful, and I've had lots of collaborators and funders, and of course my family who's put up with me so far. With that, I'll just take questions. Yes, Samir?
So one aspect you didn't discuss is like a lot of the data that you're dealing with probably has errors in it.
Yeah.
Is there a process of feeding the data before your software takes over, or how you deal with that angle?
Yeah, so the clustering is one example where the clustering is in part necessary because of the errors in the data. And through the clustering process, you can realize, well, I only saw this sequence once. When looking at 3,000 samples, there's probably an error and I can remove it. Right? So also when you see many similar things, they're probably not errors. So there is a fair amount of that. There's a lot of work in error correction of various kinds I've tried to stay a little out of that because it's often so dependent on technology. They change it so quickly. And the last thing you want is have a student work on a project for 2 years and then the technology is obsolete and they have nothing to show for it, right? So I've been trying to stay a little more on the fundamental problems, and use other people's software to develop to correct errors up front. Yes, France?
I was wondering if you're able to in your analysis, if you could, based on the clusterings, tell when different samples came from different ones of those 4 countries. In other words, I'm wondering if there's, rather than a single global profile for diarrhea, that it's more localized.
Yes, I mean, we could see some differences, and actually, when we had to publish these results, we had to essentially be very careful that everything that we said there was correct, because we're claiming, look, all these people have worked for tens of years trying to understand what caused diarrhea, and where we run a computer program and we tell you more, right? So we did stratify by country, we did stratify by age group, because age group mattered a lot, and so on, and things that we found multiple times, those are the only things we reported. But there are clear differences, and even epidemiologists will know. For example, you know, it's in the DC area you probably will get cholera, or you can get cholera, whereas if you're in the Rockies you won't, right? It just, that organism doesn't exist there. So there are clearly geographical differences that matter. Yes, Hannah.
So you kind of poked fun at them, but you said that like you give biologists an assembly and they can tell you what, they claim that they can tell you whether it's correct or not.
Yeah.
What information are they using to make that judgment?
The biologists who claim this are probably the ones who don't actually know. So the respectable biologists, or the ones who actually really want to work, they fully understand of the complexity. But there are a lot of people who just trust too much the software and say, well, it must be correct. So.
So in a sense, it's very unlike the language case then.
Yeah. I mean, that they should know that they can't understand just by looking at the data.
Well, particularly it's a language that people don't know.
Yes.
Right?
Yeah.
People don't speak the language.
Yes, France.
Do you think the second grade core standards will give you new research directions? You don't have to answer that.
Okay, that's
Okay.
nice.
Thank you.